Okay, hello. My name is Wendy Yang, and I'm a PhD student at Carnegie Mellon University, supervised by Dr. Jen Ma. Today, it is my great honor to be here and present my recent work, UNADON, which utilizes a transformer-based deep learning model to predict the genome-wide chromosomal spatial position. So here is a visualization of how the chromatin is organized inside the nucleus, which might be unfamiliar to you. So let me explain by drawing an analogy from the more well-studied protein structure. As we all know, protein is not just a linear string of amino acid. It has the secondary, tertiary structures that closely relates to its function. Similarly, genome is not a randomly floating string inside the nucleus. It is actually folded in a hierarchical and well-organized manner, which affects a lot of crucial biological processes, such as DNA replication and gene regulation. And besides, just like the protein is interacting with a lot of intracellular components, the chromatin is also not alone inside the nucleus, as is shown in this figure. So there are a lot of other nuclear components that interacts with the chromatin. So the spatial positioning of the chromatin relative to these nuclear bodies are essential for its function. For example, the nuclear speckles shown here are enriched in the mRNA splicing factors, and gene expression level is found to be inversely correlated with the distance to these nuclear speckles. And moreover, nuclear lamina on the periphery frequently interacts with the repressive chromatin regions. And thus, studying the spatial positioning of chromatin relative to these nuclear bodies are very important for understanding the interplay between nuclear structure and function. However, the underlying mechanism, such as the sequence and epigenomic determinants of this chromatin targeting towards different nuclear bodies, remains largely unknown, and there is no current computational method that is designed specifically for studying the connections between the chromatin nuclear body interactions with the 1D genomic features. So in order to bridge this gap, we propose to develop UNADON, which connects the 1D genomic features with the chromatin spatial positioning. And we take advantage of the TSC-seq genomic mapping data, which measures the cytological distances between any genomic loci to the nuclear bodies. And an example of the TSC-seq data is shown on the bottom left corner. It can be represented as a 1D signal track. So higher values indicates closer distances to the nuclear bodies. In this work, we focus on two types of nuclear bodies, nuclear speckle, which is measured by SON TSC-seq, and nuclear lamina, which is measured by lambda B TSC-seq. And from now on, I'll just refer to them as SON TSC-seq and lamina TSC-seq. I'll use the two terms. So with the 1D genomic features and the TSC-seq data available to us, we define two major computational goals for UNADON. Firstly, we aim to predict the TSC-seq signals from the sequence and epigenomic features across various cell types. Secondly, we strive to uncover the potential sequence and epigenomic determinants for the localization relative to the nuclear bodies. Okay, let's delve into the details of the method. Here's an overview of the workflow. It consists of 3 steps: data preprocessing, predictive model training, and model interpretation. So specifically, we split the genome into 25 kilobase genomic bins, and for the prediction of each single genomic bin, we incorporate all the genomic bins within the nearby 5 megabase regions to provide the long-range context information. So for each genomic bin, we extract the sequence features represented by the K-mer frequency, which is further transformed into a lower dimensional space using PCA. And we also extract the epigenomic features, including the chromatin accessibility and 8 histone marks, which is represented by the occurrences of the signal peaks. So the sequence and the epigenomic features are fed into the neural network separately, merged into a multi-model representation, and then passed through a stack of the transformer encoder layers. The details of the transformer encoder layers are shown on the right. So the most important components of the transformer encoder is this multi-head attention mechanism, which we have seen a lot of times these days, and we know they're very powerful. So the TSC-seq signals are predicted based on the output of the transformer encoder layers. After model training, we applied an interpretable machine learning technique named integrated gradients to compute the feature importance scores. So I would like to highlight 3 key features that contribute significantly to the performance of UNADON. So firstly, the pre-processing of the sequence features greatly reduces the dimensionality of the input sequences while preserving the informative features. And this allows the model to efficiently learn the complex sequence features. And secondly, the multimodal design of the neural network allows it to learn the sequence and epigenomic features separately. Thirdly, the multi-head attention mechanism inside the transformer encoder module allows the model to capture the crucial long-range dependencies between the genomic bins. Okay, that's it for the method. Let's move on to the prediction performance. So to comprehensively evaluate the predictive power of UNADON, we conducted 2 types of experiments across 4 human cell lines. So for single cell type experiments, the models are trained and validated and tested within the same cell type. And it is important to note that we adopt a cross-chromosomal testing approach, which means that we split the whole genome into training, validation, and testing set based on the chromosomes. Specifically, we use the odd-numbered chromosomes for training and validation, and even-numbered chromosomes for testing. So here are the results for the single cell type experiments. We compared our model with 4 commonly used baseline methods: XGBoost, which is based on gradient tree boosting, dense neural network DNN, which only contains the fully connected layers, convolutional neural network CNN, and dilated CNN. Among these methods, XGBoost and DNN cannot incorporate any context information. CNN and dilated CNN are commonly used for the sequence-based predictions. So on the left is the Pearson correlation of the predictions. As we can see, across all the cell types and nuclear bodies, UNADON shown in blue achieves comparable or superior performance compared to all the baseline methods. And on the right, we show an example of the predicted versus actual signal tracks. As you can see, they match pretty well. The highlighted region illustrates that UNADON accurately predicts the cell type-specific patterns. And more importantly, even the small variations in structures, like the tiny peaks and valleys smaller than 1 megabase shown here, are predicted correctly. And for cross-cell type experiments, one cell type is held out as the testing cell type, and the model is trained on the rest 3 cell types. And still, we use the cross-chromosomal testing approach here. So this is to evaluate the generalizability of the model to unseen cell types. So we included an additional baseline method named average, which is shown in orange here, which simply utilizes the average signal of the 3 training cell types as the prediction. It turns out that the average baseline works particularly well for the prediction of the song TSC-seq, which is highly conserved across the different cell types. But still, in this case, UNADON achieves slightly better performance. But the advantage of UNADON over the baseline methods becomes more evident in the prediction of the lambda B TSC-seq, as is shown on the bottom. And across all the cell types, UNADON achieves a median Pearson correlation of over 0.75, which indicates that UNADON can predict the TSC-seq signals in an unseen cell type with moderate accuracy. So on the right, we show an example of the cross-cell type prediction of the K562 lambda B TSC-seq. So the first 3 tracks are the training cell types, the bottom 2 tracks are the predicted and the actual signals. And we highlighted this region with a unique pattern that is specific to K562. And as you can see here, UNADON accurately predicts this distinct pattern, which indicates that UNADON is really learning some fundamental connections between the input genomic features and the spatial positioning, rather than just memorizing the training data. Okay, that's for the model evaluation. And another question we want to ask is what features are actually important for the prediction. So we investigated the contribution of different genomic features by applying an interpretable machine learning method called integrated gradients. We summed up the genome-wide importance scores for each genomic feature across all the experiments, and that is summarized on the left. So each row represents one experiment, and the different color bars represent the contributions from different genomic features. So The first observation we can make based on this plot is that sequence in dark red is the most important feature for the prediction. As you can see, it takes up a large proportion here, followed by ataxic in red and H3K4 monomethylation in orange here. Besides, the distribution of this feature importance scores varies significantly across the cell types. For example, if you look at the contributions for lambda BTSA here, you can see that sequence is much more important for the prediction of the HCT116 than it is for HFFC6. Besides, if you compare the contribution for SONG versus lambda BTSA6, you'll see that epigenomic features are more important for the prediction of lambda BTSA6. And if you compare the same setting of the single cell type versus cross cell type experiments, you'll see that the cross cell type models tend to rely more on the epigenomic features. And this is because the sequence features are the same for all the cell types. And in order to generalize well across the different cell types, the model need to take in some cell type specific information, which is provided by the epigenomic features. Okay, what we have just performed is a global analysis of the importance scores. But if you zoom into a local region on this stacked plot of importance scores, it actually reveals more detailed patterns. So as you can see, like in this region, several epigenomic features consistently appear together in their contribution to the prediction. And also in these two highlighted regions, we can observe a co-occurrence of the green like H3K27 trimethylation signals with the dark red like sequence features. So based on these observations, we hypothesize that certain features might collectively contribute to the prediction. And to quantitatively evaluate this joint contribution, we conducted a k-means clustering analysis for the importance scores. And the clustering reveals 6 distinct patterns of this joint contribution. So cluster 1 and cluster 2 has contribution from several epigenomic features. Cluster 3 is marked by this unique contribution from the H3K9 trimethylation. Cluster 4 is marked by the contribution from the H3K27 trimethylation, and cluster 5 and 6 only has contributions from the sequence features with high and low contributions respectively. And interestingly, we didn't find any cluster with high epigenomic contribution but low sequence contribution, which further confirms that the sequence features are very important for the prediction. We hypothesize that these contribution patterns might provide some insights into the different targeting mechanisms. Okay, here we revisit the stack plot of the feature importance scores and show some examples of how each contribution pattern actually look like on this genome browser. We can see some sequence-only clusters 5 and 6 here, with only the dark red sequence contributions. And we can also see some colorful clusters 1 and 2 here, which has the contribution from multiple epigenomic features. And you can also see, might be hard to see, but it's like cluster 3 here, which has the unique contribution from the H3K9 trimethylation, and also clusters 4 here, as we highlighted before, it's marked by the joint contribution of sequence and this H3K27 trimethylation. So lastly, we were curious about like what exactly are the potential determinants or mechanisms represented by each cluster. And in this section, we focus on the analysis of the lambda BTSA6 signals. A key question is where is each contribution pattern localized? To answer this question, we compared each cluster with the spin state annotation, which is a nuclear compartmentalization method that partitions the genome into speckle, interior active, interior repressive, near lamina and lamina states, which indicates the localization of the regions. So based on the enrichment analysis, we found that clusters 1 and 2 are predominantly localized in the speckle and interior active regions, indicating that these clusters might be important for the prediction of the lamina BTSA6, but they might not be directly involved in the targeting mechanism because they're really far away from the nuclear lamina. So cluster 3 has a strong association with the lamina-related states like near lamina and lamina here. So making it a very good marker for the prediction. And this is consistent with the previous findings that H3K9 trimethylation is enriched in the lamina-associated domains. And also we have this cluster 4 with the unique contribution from H3K27 trimethylation. It is not directly within the lamina, but it's located like in close proximity to the lamina states. So they might potentially mark the boundary of the lamina-associated domains. And also the sequence contribution only clusters 5 and 6 have a slight preference for the lamina-related states, which indicates that maybe some certain sequence elements are important for the targeting mechanisms. And we have conducted a more detailed analysis of the clusters 4, 5, and 6. So if you're interested, please check out our manuscript. So to summarize, we started by explaining that the chromatin spatial positioning is a crucial aspect of nuclear organization, and it is important for understanding the interplay between nuclear structure and function. So we developed the UNADON, which is a transformer-based deep learning model, to accurately predict the chromatin spatial positioning from the 1D genomic features. UNADON not only performs well in the single cell type experiments, but also generalizes well to the unseen cell types. By conducting model interpretation, we conclude that both DNA sequence and epigenomic features are important for the prediction, but this contribution is highly cell type specific. And finally, the analysis of the feature importance scores reveals some potential targeting mechanisms towards the nuclear lamina that involves several histone marks. Okay. So at last, I would like to thank the members of my lab for their support and contribution to this project. I would also like to thank our collaborators for their helpful discussions. And that's it for my presentation, and I would be happy to take questions. Do we still have time for?
We have time for one question. Thank you so much, Wendy. If anyone has one brief question, can you please come down?
Hi, very good presentation.
Thank you.
I just want to ask a quick question about your 4 cell line. It seems like 2 of them are cancer cell lines, and then 2 of them are kind of like normal embryonic and fibroblast cell lines. Do you see some differences between cancer versus normal cell lines in terms of this positioning and epigenomic?
I think that's
features?
actually a great question. We actually see like K562 and H1 are more similar in terms of like their TSC6 signals, and HCT116 and HFFC6 are more similar. This is not divided by like cancer versus normal, but it's like more based on other features, like the cell shapes and other features.